Actually, Powered, before you continue, Mark,
Sure.
I'm curious.
Yeah.
Excuse me. I'm curious as to your thoughts here. So it says, it is worth noting that we have analyzed only the two changes, so the two substitutions, one letter for another,
Right.
that are known to have occurred in exon 7 of the FOXP2 gene. But notice this, after the divergence of humans and chimpanzees. So I guess my question for you, Mark, is how do we know that those differences are the result of a substitution mutation, two of them, after the split?
Well, it's because we rely on other evidence to know that split has occurred, like chromosome 2 fusion. The simple thing that chimpanzees are closer genetically to us than they are to any of the other great apes. That coupled with other evidence,
how do you know this
between us?
How do you know these specific substitutions took? Because it sounds like you have to go infer from other lines of evidence, human chromosome 2. We could examine that for sure. But these specific changes in the FOXP2 gene, how do you know that these differences
how do we know
truly
it was a
DNA differences?
Well, we know that it's a mutation within the DNA.
But aren't you assuming? Aren't you assuming?
Well, no, we're not assuming that there's a split between apes and humans. That's been demonstrated from like a ton of evidence over our genetics, including the sort of the chromosome 2 split. The Human Genome Project has found that they are almost.
But I mean but Mark, I understand what you're saying.
Yeah,
I appreciate it. But I mean not the split necessarily, but the DNA differences that we find, the substitutions, right? One DNA letter for another. How do we know it wasn't always like that? Because aren't you assuming that all DNA differences are the result of mutations?
That's all we've ever seen between differences in DNA. Like if, for instance, you have parents that basically you have a novel mutation, that how else do we see it occur?
Right. But when
I don't see fairies with magic wands going to DNA and magicking up different mutations in things. I'm not sure how you're sort of saying it's happening here.
No, I understand that. But wouldn't you agree that the millions of DNA differences that we inherit from our parents, the majority of those are pre-existing and only a few are mutations each generation?
Well, yeah. So, I think you're talking about recombination. Is that right?
Well, like, for example, my kids would have 50% of my DNA, 50% of my wife's DNA with some mutation.
Right, but that would be pretty well random kind of thing where it's coming from. Either your sort of one ancestor or the other, basically one parent or the other.
Right, but Mark, I'm asking this: if only a few of those DNA differences are due to mutation, because you said we observe them per generation, that's true.
Sure.
In autosomal nuclear DNA, we inherit about 100 genetic mutations. Okay, that means the great majority of what's inherited are pre-existing, as in they weren't due to mutations that we've observed. They're just pre-existing now being passed on.
We can also see it in other mammals kind of thing. Like why do we have sort of the genetic similarities to say mice, for instance, but we still have these changes, and as we put the changes into some sort of system, it's a hierarchy. It's a nested hierarchy of how we're coming out compared to other mammals. We look at those things and we say, well, if this change happened here, then but you're sort of advocating that, hey, we were never related to chimps, and that was always like that. And I don't know how you can know that. It seems to be just a dogmatic assertion.
But aren't you assuming in phylogenetics?
No, we're not assuming. You're
not assuming. No, you don't even know what I'm about to ask. So you're not assuming that all DNA differences in your phylogenetic analysis are due to mutations?
What do you mean all difference in a phylogenetic mutation? Well, yeah.
Well, because
I mean,
you're
but
comparing
yeah.
DNA differences between different creatures that form a hierarchy,
yeah.
right? Right. So how do you
Yeah.
know? You're assuming that all those DNA differences are truly due to mutations over time.
Well, if everything is related and evolution is true, then yeah, that's the only way. Well,
Yeah.
yeah. And
But
there's a
if
ton of evidence for evolution where there's none for a sort of animal being created from nothing, having all of these changes within it. Like we can observe mutation happening. We can observe mutations producing
but you
vastly
didn't observe, but
Here's the difference.
but going back
phenotype
earlier, those
in-
those millions of inherited DNA differences,
well,
those
we
we
can
didn't observe
observe
from about through
mutations
mutation.
producing vastly different phenotypes in all kinds of animals. That's not even up for dispute. I don't think even you dispute that.
But Mark, my point is when you look at
Yeah.
the human genome, the chimpanzee genome, and
Mhm.
you see DNA differences, you didn't
Yes.
see those DNA differences come about through mutations. You have to
We don't
assume
have
as an
to.
evolutionist. You
We
have to
don't have
assume
to.
that they're there through mutations.
We don't have to assume anything because
You
we
do.
know that the chimpanzee and the human have a common ancestor due to a ton of evidence for it.
Okay, okay, fine. We'll go one at a time then. A ton of evidence. Pick one. We'll
Yeah.
go one at a time.
Chromosome 2 fusion.
Okay, chromosome 2. So
Mhm.
we find a very similar
Well,
signature.
no. We predicted to find a cluster of telomeres in the center of that. We predicted it first.
Did you predict the differences in the chromosomes, or did you make this prediction?
No,
after you
we
saw the
found out the
difference?
number of chromosomes, and then it was predicted if evolution is true, or really if evolution is false, then we should find the chromosomes not having any kind of fusion with them, because the only way that we can be so close genetically and still evolution is true is if a fusion happened between two chromosomes, because we do have
So
different
we
numbers.
find the differences, right? 46 versus 48. Your
Yes.
great apes, orangutan, gorilla, chimp, bonobo, they have
Yep,
48.
so we predicted.
We have 46.
Yeah.
Okay. So we discover this data, this discrepancy. So you make a prediction. Well, there had to have been a chromosome fusion at some
Correct.
point since the split to explain the difference. What would the alternative have been?
What do you mean? What will the alternative mean? No fusion and having
Yeah,
different.
so if it
different
weren't a fusion,
chromosomes.
right.
Well,
If
we
it weren't a
wouldn't.
fusion,
If it
what else
wasn't a
would it have been?
fusion, we wouldn't find the large grouping of telomeres, telomeric sequences, fragmented telomeric sequences in the middle of the genome, and we would actually find that they don't have those sequences.
How specific were they in the prediction? Did they predict how many of these
Well, I don't think
interstitial
it's. I
telomeric
don't think it's. I
repeats were
don't
there?
know. I don't know, but I don't think that's relevant. The fact that they predicted them at all and said, well, this is what we should see if these chromosomes have fused together, is enough by itself. I don't know why it wouldn't be.
Well, how large? How many sequences is a typical telomere?
I don't know. It's usually
Let me-
thousands to start
Thousands.
with.
Let me
but
ask
the
a
rate
question.
as things
Well,
get older,
hey,
sure.
T-Rock, before you do, because I'm just asking questions just to kind of gauge.
Well,
the
I
argument.
also did have a something about the thing that you said, that how do the cell repair mechanisms or the DNA repair mechanisms and the DNA sort of evolve at the same time.
Okay, well,
and so I do want to get to
I
that
don't
as
want to.
well.
I don't. We can definitely touch on that, but
Sure.
when it comes to, because you were right, a typical telomeric sequence is a telomere, the end caps of the chromosomes, are thousands of sequences long. Okay. So if you had two chromosomes slammed together, how come they didn't make a prediction as to the size, right? So we do find in the chromosome.
I don't know if they did. I don't know. I said I don't know. I didn't say
Okay, no,
they
I
didn't
appreciate
predict the size. They said highly dense, basically, which that's exactly what they found. And I know you do this thing of, but there's other not with that density and not the reversal of the sequence as
No,
well.
well, we did find the reversal, so I do appreciate those points.
Yeah, but not
We
the
made-
density that they predicted of the telomeric sequences.
Well, here's the thing: the density is
It was always about the density. It was always about Erica sort of did a video on this where you sort of say, hey, there's other sequences elsewhere, and it's the density that they predicted and the density that's important. And they've always said that, even though you say, hey, you're just making no, it's always the density. And you know, she got video of her saying prior to even talking with you, it was the density that was the thing that tipped us off.
Well, I did a response video to her, a thorough one, and it was the challenge was we don't find these telomeric repeats, right? TTA, GGG, CC, CCC, TAA.
No, it's not that we don't find them. We don't find the density.
Mark, just quit interrupt. I'm trying to be as professional as possible. So we don't find them, the claim. We don't find them. We do find them in isolation, but we don't find them in mirror form. Okay. Head to head.
And the density that they're in.
Right. Density is relative though. Yeah. The chromosome 2 fusion site has a high density compared to other locations in the genome that have instances of TTA, GGG, CCC.
Is there any site more dense?
Well, this one, the chromosome 2 fusion spot is the most dense.
Correct.
Okay? But that it'd be like saying, well, this guy bench presses 300 pounds and this other guy bench presses 500. So the guy who bench presses 300 is weak. It's not a lot of weight. Well, both are a lot of weight.
Just
300 and 500.
They predict it would
both.
Have, like,
I just want to finish what I'm saying.
You're sort of mischaracterizing it, because the chromosome 2 fusion was predicted to be the most recent, and that's why they were saying the density is going to be high, because it was the most recent. So as you know, all of these areas, it's not
high compared to other fusions that we know are fusions in other animals, like in mice, okay? Because the whole location site is only about 800 base pairs, okay? I just want to say this, then you can respond. And so Chris Roop and I, we made a prediction. We looked in BLAST in the genome, and we said, okay, let's see if we can find these internal telomeric repeats in mirror form. Head to head, TTA, GGG, CCC, TAA. So we found several, okay, but very low density. But we did find one that had high density. I will admit, not as high density as the chromosome 2 location, but still high density compared to other locations, okay? And so we discovered it in the chromosome 9 site. And this is the location. I printed it off here. You can see the location here, the query. And I highlighted. So here's the head-to-head, TTA, GGG, TTA, GGG, and then head-to-head, CCC, TAA. But nobody says that the chromosome 2 or chromosome 9 location where we found this is the result of a fusion. But what we do find is that these internal telomeric repeats are involved in gene expression, this process by which DNA directs the synthesis.
You're sort of going off the point, Donny. You're sort of moving to another thing.
Okay, go ahead, respond.
So my point is that the other one was 400. This is tiny. This is not even
the other one's tiny too.
Well, no, not compared to this one. This one's like what?
Well, compared to real fusions in other animals, it is.
Okay, yeah, but the one that you said mice, that's where we've intentionally fused their chromosome. It's like just happened. Like, this is the thing. You're sort of comparing us fusing chromosomes out. Well, it's not as large as that one. Well, no, of course it's not. It happened ages ago.
Mark, you're the one that said a telomere comprises thousands of sequences.
No, I didn't. No, don't stop misrepresenting me. I said,
okay, so how many sequences does a telomere?
Thousands at the start, but it degrades over time, as you're well aware. It can go down to, and when it hits 0, that's when the cell dies, as you're well aware. So it can be anywhere from nothing to 1,000 or 1,000.
The issue is the number.
As it gets older, it removes those sequences, and that's what causes the cell to die. So you know this, Donny. I know you know
it. I've written on
something
it, and I've
being like 400 is roughly where we'd expect it to be.
Yeah, but the thing is, you're saying that after the fact. But a major paper. Well, it's like when I pause, then you think you can interrupt. I'm almost done. Okay, so a major paper, I appreciate the passion, Mark. So a major paper on this issue, they asked the question, why is the area so degenerate? They didn't say, oh, the area is degenerate, it's small. This is what we predicted. This is what we expected. No, they asked the question, why? And then they put forth several possible hypotheses. So no, it is a lot smaller than they expected it to be if it was a few. You're saying it's a fusion, but it is smaller compared to fusions in other animals. In the human genome, yes, and it's right here on the screen. This is the fusion site, right? TTAGGG. Here's where it connects. TTAGGG, and then you get into the CCCTAA, okay? 798 base pairs. You can compare that with the chromosome 9. Yes, it is more dense than the chromosome 9 spot, but the chromosome 9 spot is still dense. It's just not as dense as the fusions. So the question is, why are these areas
it's not
dense?
Like, come on now. It's how many repeats is it?
In the chromosome 9?
Yeah.
You can count them right here. I think it's
what, less than 10?
I think it's about somewhere between 10 and 15.
Yeah, I don't like compared to the other one. It's not even half.
Well, there's not that many
size.
here either. Look at 1, 2, 3, 4, 5.
So, but the thing is, and I'll say this lastly, whether or not it's more fragmented or more degraded than what they expected is aside the point. It still doesn't address how they predicted that point or roughly that point in the chromosome to have that density of sequence. And you've sort of said, well, did they do that after the fact? No, they didn't do that after the fact. I think that's a bit ridiculous, to be honest.
Well, I would challenge you to show me where they predicted how dense it would be. I want to see a number.
I don't have
because they did ask, why is the area so degenerate? How come they didn't say,
sure.
This is what we expected? It's 700 to 800 base pairs.
Well, sure.
And that's what we expected.
Well, it's because, we make predictions, and based upon those predictions, we learn a lot of stuff by, saying, hey, what they predicted about the density was correct, but obviously it was more degraded than what they expected. But that doesn't mean they're wrong about the prediction that there should be a dense region of telomeric, loci in the chromosome 2 at exactly the point that they were, or maybe not exactly, I think I'm overspeaking there, but at roughly the point where they expected it to be. And this isn't apologists or creation ministry going out, or Discovery Institute going out and discovering this thing. This is them after the fact saying, oh, we're going to try and disrupt it by saying, hey, it wasn't this or it wasn't that. I don't see them making any predictions for.
Well, we did make a prediction and it was fulfilled. We predicted, are we going to find any instances in the human genome where we have the telomeric repeats TTA, GGG, CCC, TAA in a head-to-head fashion that is somewhat dense? We went into that without knowing, and we discovered it in the chromosome 9. So my question to you, Mark, do you believe that the chromosome 9 is the result of an ancient fusion because they have those repeats?
It could possibly be. I don't know. I'm not a geneticist. I'm not a biologist. It could possibly be. I don't know.
Okay.
But so if it is, it's hang on. If it is, it is much, much older than the chromosome 2 fusion. Like probably, mammals some point in the past, a long way away kind of thing. I don't think it would be something within the recent few million years, definitely.
So if I could show you a source or two that concludes the chromosome 9 is not the result of a fusion, yet it has similar characteristics and traits to what we find in the chromosome 2 location, but I admit it is more dense than the chromosome 9 spot. Could therefore the chromosome 2 not be the result of a fusion?
Like I said, I don't know. I'm not a geneticist, but the fact that they predicted it at all, and the chimpanzee genome is so close to ours, it's incredibly similar, and it's more.
But I mean, we're supposed to be 50% similar to a banana plant.
Well, I don't know about that one. Have you done
we're also 90% similar to tobacco.
Well, we just reviewed a video by Rationality Rules where he made the argument that we're 96 to 97% similar to chimp.
That could well be true.
And then he also said we're 50% similar to a banana. So that makes sense. Yeah, hold up a banana and then look at a picture of a chimpanzee.
Okay, so that is basically an argument for incredulity. You're just appealing to incredulity to say, hey, that can't possibly be true without any reason why it can't be.
Well, because the argument from similarity, which you're looking at, okay, is largely agnostic, because you would say the similarity is there due to common ancestry. And I would
Well,
say
no.
it's due to common design. So we'd have to look at something that would discriminate the models.
So hang on a second. But so are you saying that chimpanzees are more similar to other apes like orangutan, gorilla, and things like that?
No, well, it depends on what you're looking at. But whole genome wise,
what do you mean? What
humans are most similar? Well, if we were to look at the Y chromosome, then the human and the gorilla Y, they're more similar to each other than either are to the chimpanzee Y. So it depends on what you're looking at.
Well, I mean over the whole thing, yeah.
Right, yeah. Oh, yeah, the whole thing. Humans and chimps are more similar than humans and orangutans or humans and even gibbons. But you can just stand afar and look at a chimp, look at a human, look at an orangutan, and it's pretty clear that we share more in terms of morphology and anatomy, and therefore the underlying blueprint would be more similar as well.
But we also discussed that wasn't the case either. That was the whale and the shark argument again.
I mean, Mark, are you surprised?
Look the same. That
are you surprised that? Oh, I'm sorry. Are you surprised that we share 50% with a banana plant? Because we know we look nothing like a banana plant, and yet we share 50% DNA with a banana plant.
Well, we don't have to. That's the thing. We don't look at you know, whether somebody looks like their parent in order to see if genetically if they're related. Like, for instance, somebody with a lot of melanin in their skin and marries a person who is does not have a lot of melanin in their skin, they might produce a offspring with very white skin. But we don't look at the daughter or the son and the person, the black parent, and say, hey, they look nothing alike, they can't be related. That is not how genetics works in the slightest. So I don't know why you would say, hey, I look at a thing and it seems like this, therefore that's the case.
Oh no,
I don't know why.
I'm just pointing out I'm acknowledging the agnostic point of it. Just because the similarity does not equate design, or else convergent evolution wouldn't exist. So even the evolutionary community will look at two very similar creatures and in many instances admit that those similarities are not due to homology, they're due to convergence. Okay, so I'm just pointing out that the mere reality of similarity does not equate relationship. Well, we got to dig deeper than just saying, well, humans and chimps are 97% similar, therefore they go back to a common ancestor.
I think you're being a bit dishonest here, because I've just looked it up, and if you look at protein coding genes, there's a high degree of similarity. That's where the 50% comes from. But you look at the entire DNA and it's only 2%.
Okay, so you're 96 to 97% similarity with chimpanzees then. Is that only protein coding genes?
I don't know. It's 90 something. I don't remember. 90. I can't remember the exact number. It depends on how you actually do it.
I only ask you that because the same source that says humans and banana plants are 50% the same also says humans and chimps are 97% the same. So maybe that source is only looking at protein coding genes in both examples.
I don't know. I think that even if that's true, and it's just the protein coding genes.
Well, the point is then the similarity between humans and chimps would be lower.
If it is just protein coding, then we are almost twice as related to chimps than we are to a banana.
Well, we'd have to answer the question if we're only looking at protein coding genes.
Sure. Why don't you get that for me? Why don't you answer
to me, it does, whether humans and chimps are 99, 97, or 50% similar. Okay. Similarity doesn't equate relationship. And so I would want to look at a differentiating line of evidence. I think the Y chromosome is a differentiating line of evidence that I have up on the screen. I mean, you're asking earlier about that, and look, human and yeah, so I appreciate
you have a paper for that.
Yeah, you can.
Is that
right here.
Paper?
The Y, excuse me, the Y chromosomes of the great apes. I could pull up the new paper. Eric and I just had a debate on it for 2 hours. There's a new 2024 paper that actually has the human and chimpanzee Y chromosomes even less now. It's at like 25% similarity. It's amazing. And so this
over the entire genome.
Well, just the Y. Well, here's the thing: the Y chromosome's non-recombining. Would you agree?
Yes, but it actually exhibits a high level of mutation.
Right. Yeah. So how come if it, and I agree with you there, Mark. And so how come there's such uniformity then in the human Y chromosome if it exhibits such a high mutation rate, which again, I agree, it's about 1 to 3 per person on the paternally. How come there's not more variation worldwide in the Y chromosome?
What in what sort of time frame are we talking about here?
Well, let's say you go back 100 to 200,000 years in the out of Africa scenario, right? Okay, so you have people groups split off. They go their separate ways. They become isolated from each other, and they continue to accumulate independently mutations in the Y chromosome. Well, within 100 to 200,000 years, shouldn't there be a greater level of divergence in people groups around the globe in the Y chromosome?
No.
How come?
What do you mean, how come?
Well, if they're split for thousands and thousands of years, and yet they're
it's non-recombining. You said it yourself. It doesn't recombine.
Yeah, but it's accumulating DNA differences due to the high mutation.
Yeah, but the mutation rate isn't that high to make that amount of difference in such a short amount of time. Like, you're sort of assuming the mutation rate is just wildly out of control.
Is 100 to 200,000 years a short amount of time?
In the great scheme of things, yeah. Like, we're talking about billions of years here. It's not an incredible amount of time. Like, you're just thinking on this sort of human scale of, well, I lived for 100 years.
What's the size of the Y chromosome, though, Mark? What's the size of the Y chromosome?
I don't know. Why don't you tell me?
I think it's like 90, maybe someone could Google. I think it's about 90 to 95 million DNA letters. So that's a lot smaller than your autosomal DNA. So yeah, it would accumulate change a lot faster than if we were looking at autosomal nuclear DNA.
And it does. Just not the
so there should be a greater
that you're sort of trying to suggest.
My whole point is there should be more DNA variation in human male Y chromosomes around the globe, but it turns out we have low genetic diversity. Any two male Y chromosomes on the planet are nearly identical, but yet you bring in the chimp. Notice this, Mark. You bring in the chimp Y, and all of a sudden we have massive differences. When you look, it's half the size. Look at this heterochromatin here on the human Y. It's not even present in the chimp and bonobo Y. I mean, what happened to the heterochromatin? It was just all lost.
So you're comparing like 200,000 years to 5 to 6 million years.
So you think within 6 million years since the
possibly you could actually provide something to say that it's this wildly out of control thing that we should not see this. It's kind of speculation on your part that you're just sort of saying, hey, it leads to a creator, and I don't think that's the case at all. But you haven't made that case. All you're doing is saying, hey, there may be something in biology that we're unaware of, therefore God. All you're doing is God of the gaps. That's all
Mark,
you're doing.
I do understand your concern.
But I do have something to say on that, and I brought it up earlier, that thing where how did we develop the mechanisms for repairing DNA?
Okay, fine. Hey, let's land the plane. Okay, let's land the plane on this topic, then we'll move to that, okay? Because we are kind of getting over time at this point. I said 4 hours.
And so this paper that you did, why?
No, wait, I want to land the plane on the Y chromosome. I'll give you the last word. I just want to say this, okay? In light of what you said, okay, I would argue that this demands separate ancestry and independent creations, because the evolutionists, and here's my assertion, okay, the evolutionist lacks a convincing explanation and a sophisticated mathematical model to show us how the human and chimpanzee Y chromosomes can only be about 25% the same since the split, because those are massive difference. I mean, you're going to have to explain where, when, why, how all of that heterochromatin was lost, or how these genes were gained, or these gene families were lost, so on and so forth. So you guys, to have an empirical model, okay, you can't just attack the creation model and say, it looks like my model is true. No, for years to be empirical, you're going to have to demonstrate how these differences can accumulate, Mark, in 6 million years. Not just say that they did, but show us how. Go ahead. Have a final word on that issue.
Yeah, so this is sort of just attacking. I'm sure a sort of evolutionary biologist could actually answer that question. It's just I'm not an evolutionary biologist. But the mutation rates of the Y chromosome, it's all accounted for. That paper that you're talking about puts it down to evolution and sort of retaining stable sets of ancestral single copy genes, and it does explain it. It's just you're pointing out and quote mining a paper, sort of, you know, the studies of Y chromosome diversity in other great apes are relatively undeveloped compared to those of humans, but nevertheless have provided insights into speciation, dispersal, and mating patterns. So we can even find mating patterns in how these chromosomes are actually sort of where the mutations are happening, and follow the Y chromosome. It's just you choose to cite the paper and ignore the conclusions of the paper. So our understanding of these sequences from things like population histories, male bias behaviors, mutation processes, it's all explained, but you're sort of saying, hey, I'm incredulous about this, so I'm going to compare, you know, 200,000 years to 5 to 6 million years, like multiple times the length of time. You know, it's just sort of incredulity. And no, you haven't actually come up with any kind of explanation to how this happened and the process in which it happened, besides God done it. That does not explain anything. It's not an answer. It doesn't explain a thing. It's just an excuse, because you can't actually provide any kind of process that would allow this to happen.
So the issue is the evidence is missing. So for mutation to provide the gap, data in the gap from what Donny is saying is between, you know, chimpanzees and humans, is the same issue that I was bringing up earlier. There is a gap. So how do scientists explain it? They say accelerated evolution. They try to come up with this way to you have the data that has to exist somewhere in the genomics of species, but if it doesn't exist, where is it? And if mutation is the only thing you can rely on to get there, then how can you get there in such a short amount of time? And where is the data that you got there?
Not a short amount of time.
By the way,
not
this is actually
not a short amount
in the fossil record too, because during the fossil record, we have the Cambrian explosion, and then after that before that we had the Avalon explosion, and then after that we had a mass extinction, and then that had an explosion of life after that, and then after that we had an extinction in Mesozoic, and we had an explosion of life after that. So evolution is constantly catching up and constantly accelerating. It's not taking millions of years.
I was going to get to that evolution rate are not the same uniformly. And you sort of saying we don't have evidence of mutation. I don't even know where to go with that. We obviously have evidence of mutation and different rates of mutation to boot.
Hey,
so I have no idea
Mark,
why you would sort of say, excuse me, I'm just in the middle of speaking. You know, you've
been speaking for a very long time now.
Mark, I was told that I would have the final word, but obviously that means nothing. Obviously,
yeah, Mark, I'm going to jump in, guys, because I did say I'd give you the final word, but Mark, and you're still going to get it because I'm going to ask you one question. I just didn't know the final word would be 3 minutes of saying that. Oh, I'm ignoring the paper. I'm ignoring because like everything you just said could now, right? You'd have to admit you said quite a few things that could result in a 2-hour debate. But for sake of time, for sake of that, I want to ask you, because your argument about mating patterns and sperm competition, which I'm more than familiar with. Okay. Here's the newest paper in 2024. All right. And so they've compared in a very sophisticated manner. They've compared the Y chromosomes of all the great apes. So if you look here, okay, this visual, you take the chimpanzee. This is how you calculate it, right? You go chimpanzee to chimpanzee on the Y, 100%. Of course, that's expected, right? Then you go chimpanzee to human, it's 45.1%. But if you take human, because not all of it aligns, okay? So if you go human to chimp, now it's only 26.2%. All right, so huge difference. If you go human to gorilla, or let's see, gorilla to human, Okay, 23.2%. So you could see some significant differences. So my question here, with what you said, the mating patterns, the sperm competition, you're happy with those explanations to account for these differences? You're pretty you're satisfied with that?
No, I would like more information about it. I always do want more information about it. But just because we sort of say, hey, here is the theory and this is what we understand about it, doesn't mean that they have to explain every tiny thing. Like this is the absolute the disparity between the two sides. And I want to really point this out. I'm supposed to explain every single little nuance of biology and every single tiny little nuance of exactly what happened in the process. What have you explained about the process of creationism? The process. Explain it.
Well, we made very specific predictions. The Bible claims to be the
not
history.
The process.
Are you saying the scientific process is not?
Let me ask you a question.
Well, there's no process. You can't explain a process. It's basically the same as, you know.
Well, the scientific model is based on making testable and falsifiable predictions, which we've done in terms of validating separate ancestry. Okay.